Hey, Mr. B here. This is a video about transcription and translation, and I'll touch upon replication. Um, One video to watch which ties in a lot with my video as well, so if you watch mine and you still want to get a little more information, I'll have a link for Mr. Anderson's part 2. We had already watched DNA and RNA part 1. The part 2, which is a lot of what I'm going to cover, is going to be in this red area right here. Um, and I get in a little more detail on some things that Mr. Anderson doesn't cover. Uh, One thing about DNA, it's a double helix, right? You got the complementary base pairing. RNA doesn't have that, so this is a pretty solid visual. It's showing DNA. It's showing how you've got complementary base paired. RNA single-stranded, huge difference. As you probably recall from biology, there is no thiamine like you'd have on DNA. Instead of thiamine, you have uracil, okay? And it's the that complementary base pairs with adenine, so it's AUGC. Those would be the complementary base pairs. That you probably remember from biology. If not, you remember it now. And then the other difference is RNA has a ribose sugar, which is why it's called ribonucleic acid. And that ribose sugar, I'm going to circle my mouse. See where I'm circling right there? That OH, look over at DNA. It doesn't have that. And so this has an additional hydroxyl group right there. And that's what makes it a ribose as opposed to a deoxyribose. Right. Now, DNA replication. I'm not going to go into too much detail on this for this video. Just want to remind you that replication is where if we're going to make copies of ourselves, if we're a cell, we want to make copies of ourselves. That's where replication comes into play. A couple things to point out with replication is that here's my 3' end, is this strand that I'm going to be highlighting right there. There's my 3' end, and here's my 5' end right here. So what happens is first you've got, this little wedge here represents helicase. You've got helicase and some other proteins and some other enzymes that are involved in basically unwinding and unzipping. And I'll cover that later in another video. But, so you've got this helicase, which is breaking the hydrogen bonds between the complementary base paired nucleotides. And then what you have is, here, let's see if I follow this. So that would have been the 5'. So this would be my 3' end right here. So what it does is the DNA polymerase makes a copy of that DNA, or makes the complementary base pair from the 3' to 5' end, no problem. It's just straightforward. If you go from, because we split using the helicase, now what we have over here at this end here is what they call the 5' end, building from the 5' towards the 3' end. That's more complex. And that's where some other enzymes are used here. So you got this RNA primer, the DNA primase, DNA polymerase again, and ligase. Ligase is what that does, is that basically all these things allow you to what they call backstitch. The ligase completes the phosphodiester bond between the phosphorus backbone. So that's what this is indicating here. Is there's a break. The ligase is what allows that phosphodiester bond to form. And then if you notice on this visual, is it's still showing that the complementary base pairs aren't hydrogen bonded yet, and that's where the DNA polymerase comes into play. So the key thing is that this is just a little more complex than going from the 3' to 5' end. Alright, again, replication is something that we're not going to worry about for today. What I want to focus on is transcription and translation. Now, central dogma is where you're taking DNA to RNA to protein to phenotype, which is the physical characteristic of what we see. Hair color, skin, whatever, anything that we can physically see. In the case of our lab, we had the green fluorescent protein. Um, there's 3 steps, whether you're a eukaryotic or prokaryotic cell, and you have the 3 steps, which this visual shows them really nice. Step 1, initiate, and then step 2 is elongation, and then step 3 is termination. And you'll notice that you've got this thing called the promoter, and then the terminator right here. And so the promoter is just basically a start point. And what this RNA polymerase, here's an enzyme for us. Put that on your enzyme list, RNA polymerase. And what happens is you start making your RNA transcript, as they call it. And as this polymerase moves down the chain, notice where it's making its copy. It's copying from the, it's building from the 5' end to the 3' end, which means that it's copying only the 3' strand, right? So because if it's making the 5' end here, it'd be making the opposite, whether it be anti-parallel. So it's going to be it's copying from the 3' end, and as it goes along, elongates, and once it hits the terminator, it stops making that RNA from the DNA, and then our polymerase goes off and cranks around in our cell and waits to make another copy. Here is the difference, though, between prokaryotic and eukaryotic transcription and translation. Let's focus on the prokaryote first. What happens here is, one thing that is of importance, just to highlight again, is that the DNA in a prokaryote is circular in nature. The chromosomal DNA is circular in nature, longer, so it folds upon itself. And then there's also the plasmids, which are circular but very small compared to chromosomal DNA. In a eukaryotic cell, it's the DNA is not circular in nature. It has a distinct beginning and an end. It's very long, and that's what this visual is sort of showing there. Okay, so that's one thing of importance. One other thing to notice is, well, as you know, is that a prokaryote is very simple cells in terms of its operation compared to a eukaryote. So since it's less complex and it doesn't have a nucleus, the transcription and translation occurs simultaneously. And this term is called coupled transcription and translation. One other thing to keep in mind is a prokaryote is typically something that has a short life compared to a eukaryote. You know, how many 80-year-old E. coli cells do you know? None. Yeah, so where that would become important is, geez, if something's going to live for a very short period of time, you want to have a process that's pretty quick. And so this eliminates a lot of steps that the eukaryotes go through. And also, whether the cart came before the horse, or I don't know which came first, chicken or the egg, but one of the results is that you have a lot less junk DNA in a prokaryote. The numbers vary, but just for a ballpark figure, 15% or so of this DNA is junk DNA. In other words, it doesn't make a protein. It may make something else, or we don't know what it does yet. One other difference with a prokaryote, the initiation of transcription at promoters involves the recognition of a promoter sequence by RNA polymerase. Okay, so basically, hey, I want to make a copy of, or I want to make a protein, right? And so that means we're going to initiate transcription. So we're going to have to find a promoter. And what that means is it's going to go over here and it's going to look for a specific recognition of a promoter sequence by DNA or by RNA polymerase. And that varies a little bit over here. We'll come back to that. One thing I've seen I've read that only one type of RNA polymerase is needed for transcription. Then I was on a pretty high-end site and I was seeing that there's two. Um, Regardless, there's not too many types of RNA polymerase needed for transcription, okay? So that would be this little guy right here. Notice the things that are the same between a prokaryote and a eukaryote. They both go into a ribosome, okay? And that's what is going to make your mRNA. And ultimately, as it goes through the ribosome, we'll get into the details of that in a second, the polypeptide is the protein. So this is what makes the protein. Hey, make the protein here as well, but here we have a nucleus. So that brings us into the difference. Obviously, the big thing about a eukaryote, which that eukaryote means with a, I think in Greek or Latin or something, is like with a nut. And this, this is back in the day when they were first just looking at cells under microscopes, which are high tech back then. And they could see this, this thing that we call the nucleus. Well, notice where the protein synthesis occurs. It occurs in the cytoplasm, not in the nucleus. What happens in the nucleus is you have transcription, but there's multiple processes before you get to translation. Again, look at prokaryote. Transcribing and translation all in one shot. Here, transcription, translation, multiple processes. Okay, so here, big thing, transcription occurs in the nucleus for a eukaryote. The protein synthesis occurs in the cytoplasm. And you've got the transcription for a eukaryote. There's a lot more controls required because it's a way more complex cell. And so there's different ways that transcription is regulated. There's different levels of repressors and activators. There's the chromatin structure, which is just physically, your DNA and how it's all wrapped together. That structure itself can also help regulate transcription, right? The initiate. So let's compare. I was talking about how these two are a little different. The initiation of transcription at promoters involves a complex of proteins with the RNA polymerase. A little different than before. Here, prokaryote, we didn't need a whole bunch of proteins. I think I took the visual off of here, but I know that when you're on the, if you watch the video with Mr. Anderson, he's going to show, he's going to have a visual where it shows a bunch of blobs on his visual, and you're not going to know what those are. That's showing those complex of proteins that work with the RNA polymerase. And also, you can find that on PH School site, which we'll be going to in a second. Okay, so mRNA, that's there's pre and then there's this mRNA. I'm going to get into that in a second. Well, why is there a pre-mRNA? Well, you have these things called introns. I'll show you a visual in a second. But the RNA has to get processed. So here's your mRNA. It got processed to remove introns from here. It also had to add a cap at the 5' end, which they call a promoter. And then you had to generate the poly A tail at the 3' end, which is our terminator. That tells it, hey, stop the transcription process. And let's see, cap structure, poly, or why they have them there. You need that to export mRNA into cytoplasm. It needs, you need the cap to help initiate. You need the poly A. Well, actually, both of them really are needed also for the stability of mRNA. You don't want something that's going to be basically chemically falling apart as it's going through this process. So one of the big things with eukaryotes is, and it varies from species to species, but in general, junk DNA, and I use this term junk, it's not like really the best term, but it is what it is. That's just, that really means that's a chunk of DNA that doesn't code for known proteins. So in humans, this number varies, it depends where you search, but between 90 to 98% of our DNA does not code for the production of proteins as we know it. A lot of that DNA we don't understand yet. Basically, when you hear the term junk DNA, the best way of thinking about it is the amount of DNA that we don't understand what it does yet. And last but not least, where I set up here with the prokaryotes, there's one type of RNA polymerase needed for transcription. There's a bunch that are needed for eukaryotes, and I think it's actually 4. I had 3, but I think I found that it was 4. It really, this stuff, any of this stuff, transcription, translation, replication, what you're learning both in high school biology or AP, it's way more complex than what we're pitching. We're just trying to get you the basic ideas so that when you take this, if you take this in college, that you'll have a good base to then take on the more complex nuances. All right, so that would be why sometimes I'm finding some different answers here. But as you notice, I've included all my references, so if you want to check. All right, so the one thing about this visual that stinks, we've talked about, geez, if 98% of our DNA is junk, well, how in the heck do you get out, do you remove the introns? Introns being that junk DNA that we don't need to make a protein. And so here's a better visual. Okay, this would be how a prokaryote goes from, here's your promoter, here's your terminator, here is this big long strand of DNA, and this is be the chunk of DNA that we want to make a copy of RNA because we want to make a protein, okay? So that's where your RNA polymerase would go along and copy through transcription, and then translation will make a protein. Now, it's a lot different with a eukaryote because, like I said, there's this junk DNA, and the junk DNA is where what you have is, okay, again, chunk of DNA, you got to have a promoter, you've got to have a terminator, but the difference is you've got this junk DNA, which is kind of this lighter pinkish color. Those are called introns. That intron would be the junk DNA that we don't want. That's not going to make a protein, so we need to splice that out. And so that's why you have pre-mRNA. You have your cap, you have your poly A tail. Why is it poly? Because there's 5 A's on there, for A being adenine. And so what happens is there's a process where you splice out the introns. This process is a little more complex. There's some visuals which we'll get into in a second here. And again, it gets way more complex. If you want to dig in, you do some searching, you'll find the stuff that are teaching in college is pretty fascinating, but way more complex than we need to worry about. So pre-mRNA is processed in the nucleus to remove the introns and splice the exons together so that you have translatable mRNA, meaning that you can make it into a polypeptide protein. Okay, so now let's take a look at this site here. Control, again, this is all on PHS website. Like I said, what happens with these introns is you feel free to go through all the way from the beginning. Here it goes right from central dogma. It works through everything that we've talked about. And I'm going to use this site here. Like I've said, I'm going to use that for sources of questions, self-quiz at the end. Go ahead and take it. That's where I'm going to look for quiz or test questions, that is. So here is the processing of an intron, how you get it out. This is one of those, I forget what the heck they call these things. I forget what the SNRMP stand for. I know it's on here somewhere. Oh, small nucleoprotein particles. Whew, that is a mouthful. Like I said, it gets more complex. But just so you know, you got to have some more proteins that are going to help fold up this intron. That's what allows it to pull that intron out, the junk DNA as we'll call it, splices the exons together, and now you have your processed RNA. So you went from pre-mRNA to mRNA, which is ready to be made into a protein. So, go through the site, and basically, if you can answer all these questions on this, and as you're going through, if you know all these, you're in excellent shape, not only for this class, but also for AP. A lot of this stuff that we're doing here is the same thing that they do in AP biology. Alright, so let's Alt-Tab back. So we've gone through translation a little bit. Here is a little bit better visual because what we've talked about is basically what a lot of it's been talking about is this mRNA. We really haven't talked about how we end up with a protein yet. And that's where this visual comes into play. Again, this came from that same website I was on, and I'll I'm sorry. This is a visual that I got from this website. You can find this. This comes from, this might even be in the school's textbooks. What this is showing is, okay, so here's our mRNA, whether it was a prokaryote or a eukaryote, doesn't matter. All right, it's mRNA, and what it's going to be doing, it's going to be feeding in, that's what these arrows mean, it's feeding into a ribosome, and what you have is tRNA. And what the tRNA does is a tRNA is cruising around in your cytoplasm. It's grabbing an amino acid, which remember you always hear amino acids are the building blocks of proteins. Yeah. So the tRNA snags whatever the appropriate amino acid that it needs, and then that goes into this what they call the larger subunit of ribosome. I've got a visual a little bit later, which is pretty good. So that comes into the A site, this A site and the P site, and this the visual I'm going to show you in a second shows this a lot better. But basically, the tRNA is what you've got your codons and your anticodons, and I think we'll be seeing that on the visual coming up. But this is, in essence, in the ribosome, is where we're going to read that mRNA and make a protein from there. That ultimately is the most important thing. It's, hey, translation is where I'm taking mRNA and making a protein. So let's go to this site, control click over here. Okay, and what I'll do here, again, recommend that you do this on your own. If you're a Flash player, if you didn't download the stuff like I asked you to, well, then you can watch it right here. Okay, and so here is the small subunit, and this is showing how the process, as we understand it, actually works. The larger ribosomal subunit comes on top, and that MET, that if you guys remember AUG, that's the promoter. That's what starts. That's the initiation complex for translation. You first need to have this AUG and let's see, yeah, the start code. So the start codon on the mRNA, which is this longer chain. So your anticodon on the tRNA would be the UAC. There's a couple other start codons if you remember, but let's not worry about that for right now. So you go and you click on the next concept, and it shows, okay, so now you're going to start translating your mRNA into protein. So now you're starting to build your protein, and you can see, hey, we're building right along. It's making the connections there. There's your amino acids getting connected together. Amino acids being the building blocks of proteins. So it's pretty cool. It's showing that you're going along and you're building that protein. And if I hit the next concept, it's looking for a stop codon right here, right? So here there's this big long list of proteins that have been put together. Oh, it hits the stop codon. So then there's this release factor that says, hey, okay, we've made our protein, and then all this stuff, in essence, comes apart, goes back in the cytoplasm, and can be reused for other processes. And so this is showing the whole process. Well, I get what this is showing here is that you just don't have one ribosome reading your mRNA. It can do it. You can have multiple ribosomes at the same time. I think that's a pretty important concept. They understand that you can be working the mRNA along multiple lengths. All right. And then come back to your self-quiz. All right. So those are those concepts. Again, makes this visual. You understand this. I like this visual, but I also like the PHS school site because you see things move, and it helps make more sense of all this. Now, how can I study? You know, obviously the PHS school site's pretty cool. What I did find is this visual, which does show, um, it makes, you know, it's a static version basically of the PHS school website. So, you know, as you kind of look at this, look at, you know, if you could be talking to yourself and looking at these visuals and explaining to yourself what's happening, um, that's good. And that's the skill that you're going to need in college, because a lot of times you aren't going to have good, strong visuals like that PHS school website. You'll have things like this, and you've got to be able to talk to yourself and say, okay, what's happening in this visual? Let the visual explain everything to you. And can you explain it to somebody else? Um, so again, this basically just shows what we just saw on the PHS school site. And for credit for watching this video, what I want you to do is I want you to bring in, you guys have this. You guys basically, I just took this and I just took out letters and call it guided notes. You just bring that filled in, okay? You bring this document all filled in, and I'll give you full credit. This is going to be the 20 points, whatever I gave last time. And so you just print it out, staple it, put your name on it, and turn it in when you walk into class, right? Keep it simple. Well, I hope this helped clear up the transcription translation, making proteins, which is a really key thing that you have to understand for our GFP lab. Well, thank you very much. Mr. B, checking out.